Imagine, you're a member of Drosophila melanogaster. You could also be a mouse or a fish or really anything else, and then a scientist comes over and flashes a light straight into your brain, which causes you to start grooming yourself or walking around. Optogenetics is a technique used by neuroscientists to activate a single cell type in the brain without interfering with any others. Electricity stimulates too many cell types because the electrodes activate any cell that it comes in contact with, and medication is also not specific enough. So these light-sensitive proteins in cells are produced by genes called opsins and are used in cells to extract information from the light around them. And there's a few different types of these proteins, like halorhodopsins, bacterial rhodopsins, and channelrhodopsins. So to study many cell types in neuroscience, one of these proteins will be inserted into a neuron by splicing the gene with a promoter that carries this genetic material into the cell. This allows scientists to turn a cell's genes on or off, even in cells that would not normally be light-sensitive. Optogenetics can be used in neuroscience in a few big ways. First, we can cause stem cells to differentiate using optogenetics, and in some studies, photosensitive proteins were able to reprogram human fibroblasts. Optogenetics can also stimulate a given neuron, either by driving proton pumps to charge mitochondria or by polarizing and depolarizing neurons, which allows scientists to see how a certain cell might cause a certain behavior. For example, a pair of commissural command neurons in Drosophila can be activated to cause a behavior, and when inhibited, the behavior was minimized. Therefore, the neurons are shown to be both necessary and sufficient, and you can assume that these neurons cause that behavior. In this case, grooming. As for psychiatric treatment, interneurons can be manipulated using optogenetics to see the extent of which gamma oscillations are produced in the brain, those of various psychiatric disorders. It can also evaluate how decision-making occurs, a process that might be applicable to the human mind in the future. Although most optogenetics research at the moment has been limited to neurological research, future research, and some already in progress, may prove it to be useful in many areas of biomedical research. Using light, we can make neurons affected by retinal degenerative diseases responsive to light again, making optogenetics an option for vision restoration. Optogenetics can serve as an alternative to pacemakers to amend arrhythmias in the heart, and could lower energy used for these type of treatments. In the future, devices could be developed to better target regions within the heart, as cardiac muscle is often difficult to access with light. Optogenetics can be useful in this regard as it allows for more complex and precise activation than discrete electrical signals. To conclude, optogenetics is a somewhat new idea that has become a major strategy in neuroscience research in the last 20 years. Not only does it allow us to observe how neural circuits and individual neurons control behavior in the brain, but it also allows us to differentiate stem cells and even fix retinal disease. Thank you.